Welcome to the deep dive of Key to My Research, the science podcast by the Excellence Strategy at the University of Tübingen. My name is Jennifer and I'm your host. And in this episode, Professor Dr. Juliane Walz is joining us. She's a professor for peptide-based immunotherapy. Welcome and thanks for being here today.
Thank you for having me.
Welcome. So, can you tell us a bit about your current role as a professor? Who are you and what is the focus of your research?
So I'm currently a professor for peptide-based immunotherapy, which directly describes what I'm doing. So I'm developing peptide-based immunotherapies, mainly peptide vaccines, so therapeutic vaccines for cancer patients. So I have a question directly. What is peptide-based immunotherapy? Just to give our listeners an idea of it. Yeah, so peptides are short proteins which are presented on the cell surface of all cells in our human body and also on cancer cells. And these peptides differ between cancer cells and healthy cells and allow the immune system to detect tumor cells as foreign and to destroy them. So my research really focused on identifying these peptides and using them for immunotherapy to target cancer cells.
Mm-hmm. So this is also basically the goal of your research, to develop new therapies.
Absolutely. So my professorship directly has the name of my central goal of research. That's correct.
Okay, that's good. That's very understandable.
Yes.
Okay, and what led you here to the University Tübingen or the University Hospital of Tübingen?
Yeah, so Tübingen is for many, many years now really a center for cancer immunotherapy research. This was pioneered by Hans-Georg Rammensee, who developed and discovered the interaction of these peptides with their MHC complex, which is the mechanism of presenting them on the tumor cells in 1991. And he really pioneered this whole research field of peptide-based immunotherapy, which I already joined during my MD work, so my MD thesis. And it's really work that was started on basic research and now entered the way of translation. So really the development of clinical trials to humans. And therefore, Tübingen has a really, really strong background.
A strong background because of the pioneer work here? Or what would you say is the strength of the University of Tübingen?
Yeah, so Tübingen really builds on this pioneer work and has established really a strong immunology and cancer research community, which was further strengthened by our cluster of excellent IFIT that was started in 2019. And that really enables, so IFIT stands for Image-Guided Functional Instructed Tumor Therapy, and it really merged the three main research areas that we have in Tübingen. So imaging, making a tumor cell or tumors visible for the physicians, and molecular target therapy, so which is the direct specific targeting of tumors by drugs, and immunotherapy, which is really harnessing the immune system against cancer. And I think this really strong community that is now merged in this cluster really helps us to develop novel treatments and then also directly translate them to the patients.
We'll talk a bit more about details about your research and about the fields of the cluster later. I just like to learn a bit more about your career path. Could you just give us a brief summary of the most important steps or places you have been?
Yes, sure. So I'm really started my career in Tübingen and then finally came back to Tübingen. So I did my medical school here at the University of Tübingen. I had a few stays abroad during medical school in the US and Great Britain. And then I did physician for hematology and oncology here at the University Hospital, and very early decided that I wanted to do research in parallel to my clinical work to develop new therapies for patients. And as I've joined the lab of Hans-Georg Rammensee already during my MD thesis, I came back to them and asked if I could start my research work there. Which I did, and I built a junior research group there. I then went to the NIH in Washington, Bethesda in the US to learn more about combinatorial therapies, novel models to evaluate cancer treatments. and then went back to Germany to head immunotherapy at the Robert Bosch Cancer Center in Stuttgart. And from there, I was finally called back to Tübingen to the W3 professorship that I'm now having.
Interesting career path, and interesting to sort of start off in Tübingen, come back to the roots, so to say, but keep on developing new things. So have you always wanted to be a scientist, or did you have any other options you were thinking about?
So very, very early in my life, I decided I wanted to do something with cancer and develop treatments for cancer. This started when my mom suffered from breast cancer when I was 15 years old. And this was the start. And my father was a surgeon. And it was very early clear for me that I want to be a physician. I thought I want to be a surgeon and do tumor surgery. But then I realized that I'm much more interested on the therapies that are emerging after surgery. So that was when I decided to go for hematology oncology to really learn about the entirety of human cancers.
So your career has sort of a personal inspiration. The origin is a personal motivation.
Absolutely, yes.
So let's get into the research, into the details. You are doing peptide-based immunotherapy, or doing research on this special topic. In recent years, immunotherapy has revolutionized the treatment of cancer. How has it done it?
Yes, so immunotherapy absolutely has entered now clinical routine and has really added a layer of therapy to the classical cancer treatment, which is surgery, radiation, and chemotherapy. And this was started with the development of the so-called immune checkpoint inhibitors, which I think was claimed as the breakthrough in cancer research already in 2013. So immune checkpoint inhibitors indeed are antibodies that are able to unlock the potential of the immune system, and namely the T cells, which are the strongest cells of our immune system to attack cancer. So, and these immune checkpoint inhibitors just unlock the activity of these T cells to target cancer cells. However, they do it in an unspecific way. And based on this success of the immune checkpoint inhibitors that are now approved for the treatment of several cancers, the specific cancer immunotherapies that really targeted the immune cells directly to the cancer emerged, which are the bispecific antibodies, which are the CAR T cells, and which are now the cancer vaccines that are now absolutely in the focus of research.
Mm-hmm. And could you just explain, how do cancer vaccines work? Could you just give us an example of a special case you still have in mind?
Yes, sure. So what cancer vaccines do, and you also have to always have to see it in contrast to the prophylactic vaccines that we are applying against pathogens, against virus and bacteria, is that they are applied to patients that are already suffering from cancer. And the idea is with the vaccine to train the immune system, and in particular the T cells, to detect the antigens that are presented by a tumor and then attack the tumors. So usually you go at a site far away from the tumor where you have no tumor cells and no immunosuppressive microenvironment that is usually around the tumor to train the immune system. You apply antigens or mRNA or genetic parts of the tumor cells, and you train the immune system locally.
How did Corona change the context of your research? Did it have any effects? And if yes, what effects did it have?
Absolutely. It really boosted also our cancer vaccine program. So maybe to go back a little bit before the Corona pandemic. So we had established a very strong pipeline to develop these peptide vaccines, and we had developed also a so-called adjuvant, an immune stimulator that enables the immune system to develop these very strong immune responses against the peptides. And we've done that in the preclinical setting, so in vitro models and first mouse models. However, we are not able to develop this in patients yet. And with the Corona pandemic, we had the opportunity to use our expertise in develop these peptide-based vaccines for cancer to develop a peptide-based T-cell activator against COVID-19. what we did, because we very early realized that the patients that suffered most from COVID-19 were indeed the cancer patients that were immune compromised. And some of them were not able to develop sufficient immune responses against approved vaccines. And therefore, we decided to use our expertise in these therapeutic vaccines to develop really a vaccine that enables for these cancer patients to be protected against severe COVID-19. And this we did, and we had the support, of course, from the regulatory authorities that really want to develop something to work against this pandemic. And we were able to enter very early in the pandemic a phase 1 clinical trial in healthy adults to evaluate this COVID-19 peptide-based vaccine. And it was very, very successful. We could show that it's safe. We could show that it induces very strong immune responses. And we were then directly able to enter a phase 2 trial in our target population, which were these immune-compromised cancer patients. So we were able to treat more than 100 of these patients with our COVID-19 vaccine. And again, show that it's safe, that it's well-tolerated, and that it is able to induce, even in these highly immune-compromised patients, these strong immune responses. And this directly enabled us then to follow the track to the therapeutic vaccines. And we were able to start the first trials then in cancer using our adjuvant and our vaccine peptide platform.
So the pandemic was actually a chance to bring forward your research and actually apply the expertise you had gathered until then.
Absolutely, absolutely.
And what would you say are the biggest advantages of peptide-based immunotherapy regarding other forms of treating cancer?
Yeah. So I think in particular, the platform that we are using has the advantage that we are directly analyzing what tumor is presenting to the immune system, which means we directly can wash these peptides from the cell surface of the tumor cell. So we do not predict what could be presented. We really know it and analyze it. So we could use a tumor sample from a given patient. We wash these peptides from these tumor cells and then analyze them by so-called mass spectrometry-based immunopeptidomics, which is the method that was developed many, many years ago and was now refined to be more sensitive and use only small tumor samples. And then we could use these peptides to really see what is the tumor presenting, what is also really tumor exclusive, and then use them to develop our personalized vaccination approaches.
So could you just tell us a bit more about the method of mass spectrometry?
Yes, sure. So that's really a method that in easy word makes these peptides visible for us. So it allows us to dissect from a whole bunch of these peptides that we wash from the tumor cells, each individual peptide. And when this method was developed, it was really handwork where we had to analyze the spectra that came out from a mass spectrometer by hand. And we were happy when we analyzed 2 per day. And now we have, of course, sophisticated bioinformatic methods that allows us to really automatically analyze what comes out from mass spectrometers. So we have thousands of peptides analyzed per day.
And what do you do with these results? What do they look like? And how do you continue with these results?
So these results are indeed just a long list of amino acid sequences from these peptides. And of course, not all peptides that we find on a tumor are tumor exclusive, because these tumors present also a lot of peptides that we also find on healthy cells. And I think this is also a very or a specialty in Tübingen that we have, as we are doing this approach for many, many years now. We have a huge database from different tumors and also from all healthy tissues of the human body, which just allows us to subtract this huge list from the list of an individual patient. And then what remains are really the tumor-exclusive peptides that are never found on any benign tissue. And these we could use then for our vaccination approaches.
I just have another question regarding something you just explained, which is washing the peptides or washing them off. How does this work practically?
Okay, practically, it's indeed a protocol that takes around 2 days. So there are specific antibodies that can bind on these HLA peptide complexes and just get them off the tumor cells. So it's just we distract the tumor cells and then pick the peptides with a specific antibody, and these peptides are then entering or are then applied in the mass spectrometer.
Mm-hmm. Maybe we'll just go again. Could you just briefly explain how you wash off the peptides?
Okay. For the washing off, we just destroy the tumor cells. We make them soluble. And we then use specific antibodies that bind to these HLA peptide complexes to pick them out from this whole mass of tumor cells and cell different more.
You were just talking about the personalization or individualization of cancer therapies. You just gave us a brief overview, but could you just please again explain how we can actually personalize these therapies?
Yes. So in with regard to the peptide vaccines, we really have the opportunity to analyze the individual patient's tumor and characterize these peptides from really the individual tumor cells of the patient, and then use these peptides in an individualized approach for a vaccine. So this really means we analyze the peptides, we produce them individualized for the patient, and then apply a personalized vaccine. And there are, of course, various steps to personalize. Of course, we could only personalize to the specific HLA allotype, which is comparable to the blood group of a patient. So it's an individual marker of the patient. And we could just select peptides for these HLA allotypes. That would be the smallest level of personalization. What we also could do, and this is what we also do in a trial, is a so-called warehouse concept, where we produce the best peptides that we have analyzed before in drug quality, and then do these individualized analysis of the patient's peptide, and then select out of this pre-produced warehouse the best peptides and compose them in a personalized vaccine, which makes it much faster and cost-saving than really producing each individual peptide for each individual patient.
So what is this warehouse concept, and what are the advantages of it?
So the warehouse concept means that we produce specific peptides that we have identified as the most frequent and best peptides for a given cancer entity. And we produce them prior to a clinical trial in drug quality. So they are there and just available for clinical use. And then when we have the clinical trial and have a patient included, we do an individualized analysis of the given patient tumor's peptides, and then go back to our warehouse and select the peptides that really the patient's tumor is presenting out of this warehouse and just compose them in a personalized vaccine. And therefore, we do not need the step of producing the peptides again. We only have to fill it in a final vaccine to be applied, which makes it really time-saving. So we're much faster to apply than these personalized approaches.
Because the patients really need this vaccine relatively quickly. I get it.
Yes. So it should be not taking too long as the disease is progressing, and therefore it's helpful to have the vaccine as soon as possible. Mm-hmm. But also to have the option to personalize it, that you are really sure that the vaccine that you apply fits to the patient's tumor.
And what tumors have you had success in treating with this vaccine, or what stage are we in with this research?
So some of our approaches, we are already in the stage of clinical evaluation. So we are currently looking at various solid tumors as well as hematological malignancies, which means leukemia and lymphoma. And we have also trials running for both of these tumor entities. So the trial that is the most farthest is a trial for chronic lymphocytic leukemia, which is the most common leukemia in adults. It's mainly affecting older patients that are not fit for intense treatment regimens. And it's still a disease, even there are good treatments available, it's not curable. So we always have relapses happening after the different treatment lines. And our idea was to train the immune system against these leukemic cells with the idea to eliminate residual tumor cells that remain after standard treatment.
And in what stage of the disease or illness are you still capable of applying these peptide-based immunotherapies?
That's a really important question. Of course, the best situation is a situation where you have an intact immune system. That means that your immune system and your T cells are fit and are not compromised by any previous cancer treatment or by the cancer itself. So that's one prerequisite. You need an intact T-cell compartment. And ideally, you need a good ratio of tumor cells to T cells, which means the best time point for a peptide vaccine would be a time point where we have reduced the tumor load to a maximum. So maybe for solid tumors after surgery, where the tumor is resected, or after chemotherapy that kills most of the tumor cells. So the vaccine and the immune system would be able to eliminate just the residual cells. So I think that's the perfect situation. And also that's the situation where all the vaccines trials that are now upcoming are entering in.
And how long would it take for a vaccine to show results in this therapy?
So it takes indeed a while. So it's not comparable with the chemotherapy that directly destroys a tumor cell. So the vaccine first has to train the T cells, which takes about 4 weeks, I would say. Then T cells have to expand, and then they have to find the tumor cells. So until you see effects, I would say it takes 8 weeks to 3 months.
And how can you sort of do your research in this phase while the vaccine is working? Like, do you use certain imaging, or what is your sort of role in this period from vaccination to first results?
Yeah. So what we do and what we could do also very early in this process is the so-called immunomonitoring. So we take just blood from the patient and we could measure if there are T cells that are specifically detecting the peptides that are in the vaccine. And we could really pretend is one of the peptides detected, or maybe 10 of the peptides. We could characterize the immune response. We could the intensity could be defined. And we could also decide during that process if we have to give a boost vaccination. So that really helps us. And of course, there are more and more imaging approaches developed, which is one of the focus of the IFID cluster of excellence, to help us to monitor these T cells really inside the patient. And we now have the possibility to monitor specific types of T cells, and could also then pretend, are these T cells going from the vaccine side to the tumor cells, and what is happening there. So this is an approach that is really helpful and is also a strength of our cluster.
So talking about the cluster, what other or further areas of research are you involved in next to these peptide-based immunotherapy?
Yeah, there are 3 areas in the cluster, and I am heading the immunotherapy area. And of course, this is not only peptide-based immunotherapy, as Tübingen really provides all fields of cancer immunotherapy. So we have with Helmut Sali, a really expert in the development of bispecific antibodies that really developed these antibodies in the lab, and also has then the possibility to translate these antibodies into clinical trials with now 3 approaches currently evaluated. We have a strong team evaluating CAR T cells in the cluster, improving functionality of CAR T cells. And one of our main goals for the second funding period will also be to combine these expertise, because we really believe to attack solid tumors and also advanced solid tumors, we are not able to use one of these approaches as a single agent. We really have to combine them. And I think that's the strength here.
And how do you practically combine these expertises? How do you bring it together? Do you have meetings? Do you have how does it work?
Yeah. So of course, the cluster provides the environment for these close interactions. We have meetings of the teams, but also meetings with all the different areas, which is imaging and molecular targeted therapies, where we really sit together, brainstorm, and develop novel ideas how we could reach our goal of curing cancer, developing these combinatorial approaches. And this is not only on the level of professorships. We are starting really this interaction very early in our cluster. We have a PhD program where all the 3 areas send their PhD students, and they work closely together, and we have specific programs. If PhD students from different areas join their expertise and develop a project, they get specific funding with the idea really to foster these collaborative projects.
This really sounds like a close-knitted community and lots of expertise being able to be generated. What would you say is the research you are most interested in?
Of course, it's peptide-based immunotherapy because that's my expertise. However, I really believe that the peptides vaccine have the highest potential in combinatorial approaches. And I think one really nice example of these combinatorial approaches is also our work that we are conducting with the area A, which is the molecular targeted therapies. So the drug development for specifically targeting cancer cells. And what we have seen in our first funding period is that specific cancer drugs are able not only to kill the tumor cells, but also to induce the presentation of specific peptides. And we call them treatment-induced peptides. And these peptides, of course, are really interesting to combine a specific novel cancer drug with a vaccine targeting these drug-induced peptides, so that we really use both mechanism of action that a cancer drug provides to activate the immune system and to kill the cancer cells. And I think that's really, really a strong approach for novel combinatorial therapies.
And if you look into the future, what would be sort of the best possible outcome within the next few years for you regarding your research?
Yeah, so I think the idea of combining anti-cancer drugs to improve immune system activation and clinical efficacy, is really one of my goals that I want to follow up in the next years. And I think Tübingen provides the perfect environment to reach these goals. And there are so many preclinical work going on. And we hopefully will next year also enter the first trial where we combine for the first time the bispecific antibodies and the peptide vaccine. So I think we are really far also in providing these approaches to the cancer patients now.
And what outcomes would you expect? Can you say anything about that?
Yes. So of course, the long-term goal would be cancer cure, but that's something it won't be really possible to do that for all cancers, for all patients. However, I think a great outcome would really be to develop an approach where we could really show in a large proportion of cancer patients an advance in survival, in clinical efficacy on their tumors.
Cancer is such a big disease and there's so many different forms of it. What are the cancer sort of forms you see the most within your research, or are there any forms you focus on?
Not specifically, as this peptide-based immunotherapy approach that I presented before, as we have the ability to wash the peptides from each tumor cell, we are not focused on specific tumors. However, the cluster has really declared metastatic solid tumors, which is still the tumor entities that have the worst outcome, and where a lot of the therapies that are now available do not work. And so this is really the field where we want to work on, and where we see extreme medical need to develop novel therapies for these patients.
So this is a project or idea that you would also like to realize in the future, or is there anything you'd especially like to conquer or attack?
Yeah, so one project that we are currently working on is a very rare solid tumor, which is called fibrolamellar cancer. It's just an example that's a disease. It affects the liver, and it usually affects the liver of young adults or adolescent, and without any prior liver disease. So, and in most cases, this disease is detected late. So this means when the cancer has already spread and there are metastases. So the cancer is not treatable by surgery. And indeed, so far, there is no approved treatment for this specific tumor entity. And what we've done already in preclinical work is we have developed a vaccine that is targeting a specific genetic alteration in these cancer patients. And we are currently evaluating this specific vaccine in a clinical trial. And I really hope that this will change the treatment landscape of this rare cancer where there's nothing available for these really young adults. We have one case where we applied already this vaccine to a 15-year-old girl that was in a situation where there was no treatment left, and she suffered from this fibrolamellar cancer. And we were asked if there's any possibility what we could do. And we designed this vaccine for that patient and applied only 2 doses and could show that we induced a strong T-cell responses against the vaccine peptides. And since we applied this vaccine and induced the T-cell responses, the patient had no relapse anymore. So she's disease-free, which is now 3.5 years since the last dose of the vaccine. And this is something what really drives all of us and enabled also then this clinical trial where we could now offer this vaccine to many more of these patients. And I really hope that this example in a rare tumor entity could be spread to other solid tumors, breast cancer, lung cancer, to make these vaccines just available for more patient groups.
And when can you say that a vaccine is successful? I mean, you had one case, but how many cases do you need where you get positive responses to say, we can put it out there, we can use this, this is a pioneer, basically, this is a novelty?
Yeah, so that's really an important question and goes back to really strong regulatory work that we are doing in parallel. Of course, it needs first phase 1 trials where we show safety, we show immunogenicity. So we just show that a vaccine works, it activates T cells. And then we maybe show first efficacy in these phase 1 trials, but then we have to expand. We have to go in phase 2 trials where we compare our vaccine to the standard treatment to really prove that there is an advance by our treatment. And then finally, we have to go in phase 3 where we evaluate this in large patient cohorts, also at different study sites, until we then finally could manage or could think about applying for really approval of a drug. And this is something, I think a really specialty of Tübingen that we are able to conduct these phase 1 and also phase 2 in the academic setting. Usually this is done by the pharmaceutical industry. And Tübingen really has this strong translational work that we have now. I think it's 17 trials that we have started in this first IFID funding period, which is absolutely great. But then we really need the pharmaceutical industry. So what we are also doing is to build spin-off companies that are then able to pursue the further steps of finding approval, because then it really needs expert. It needs, of course, external money to then manage an approval process for these drugs.
And this whole process from stage 1 and development to stage 3 and having a vaccine approved, how long does it take?
Also a really important question. So usually when you think in the pharmaceutical industry, I would say it's around 20 years. So it's very, very long. And we really hope as we do this step of preclinical development, phase 1 and even phase 2 in the academic setting and are very fast, we really hope to shrink down that process to less than 20 years. I could not promise how fast it will be, but I really hope that this approach of academic development, spin-off companies that are then really driven by the people that develop the drug and are connected to the research that is behind it, would help to speed up the process.
Does AI have any effect on your research, like to fasten the process?
Not the process of clinical translation, but AI is really important in the preclinical development of novel drugs and also development of peptide vaccines. And this will also be something that we implement in the hopefully second funding period of our cluster. We have 2 approaches where we want to integrate AI, which is also the development of novel molecular targeted therapies, where there's a really, really strong community now in using AI to better dissect targetable regions in a tumor cell. And we also would like to use AI. I mentioned before this huge database where we have millions of peptides in, but we have to decide which is the best one. And I think there the data set is so huge that AI and model-based learning would help us to later really look at these huge peptide lists and have the support of AI to select the best peptides and speed up the selection process.
Mm-hmm. Was that the warehouse concept?
That was before the warehouse concept. So thinking back on this washing approach where we wash the peptides from the tumor cells, and we've done that for hundreds of different tumors. So, and this built up this database where we have now these millions of peptides, but we not always know what would be the best peptides for a given tumor. And I think there, the data that we already have would help to train models to then, in a prospective way, select the best peptides.
So one big research area of the IFIT cluster is imaging. Can you tell us a bit more about this specific method?
Yes, sure. So the imaging part of our cluster is really important in two ways. It develops novel imaging techniques that enables us, on the one hand, to select the right patient for the right drug. So imaging of specific targets on the cell surface so that we really could select patients without even taking a biopsy of the tumor to decide if the patient is eligible for a specific antibody or CAR T-cell treatment or also peptide vaccine. So that is the first important part. And in the second thing, after application of an immunotherapy, this approach really helps us to monitor what is happening with a specific immunotherapy component, so maybe with the CAR T cells, because we really can now monitor them and could see, do the CAR T cells enter the tumor site? Do they enter only one specific tumor site? Is there a tumor site that CAR T cells could not enter? And the same also for the T cells that we induce by our vaccine. So as I said before, we induce them far away from the tumor. We do it usually in the subcutaneous skin of the abdomen. But then we want to know if these T cells traffic to the tumor cells and where they traffic, and if this trafficking could then be correlated to a clinical success. And there the imaging is really, really helpful. And we are now developing novel approaches to really track different kind of T cells, to also track different functionality states. So we also want to have activated T cells, not T cells that are exhausted and could not work against the tumor. And we have now many, many possibilities to monitor really these T cells in vivo.
So we've learned so much about the research of the IFIT cluster, about your personal interests and motivation when it comes to your research. So what are your interests or passions alongside your research?
Yeah, with 2 children, there is not much time because I'm really sharing my time between my role as a scientist and physician for the patients, for the research, for my staff and my team, and my 2 children. So that's always a very hard time to really do both and do both good. However, when I have time beyond, I love to travel with my family to show my kids the world. And when I did not have kids and did not were involved so much in research, I was also very active doing sports. I had 2 horses when I was a child and also during the medical school. I did show jumping, I did dressage. However, I finally decided to give up this to do research, and maybe later on I will have time to go back to horse riding again.
I was just going to say, is it something you'd like to integrate into your life again at some point?
Sure. Maybe my children will at some times find also this kind of sport interesting, and this would be a great chance to start it again.
Yes, it's actually great. From a certain age on, I think children get you to go back to certain things. As for example, I got back into ballet with my daughter. She wants to do horse riding. I'm not sure if I'm going to get into that, but it might be a possibility.
Yeah, my husband, I think, will not be happy to do the horse riding because it's extremely time consuming. I'm absolutely thankful for my parents that they did all this, so managed to bring me on this great career track, and also managed to enable me this really great activity.
I'd just like to get back to the University of Tübingen. What role does the University of Tübingen play for you as a scientist?
The University of Tübingen really provides me the perfect background to do my science. And also starting as a young physician in clinics and wanting to start a scientific career, there are so many great opportunities to support young researchers. I entered the Fortune program, that it's an internal program that supports first staff and funding to start research projects. There are also a lot of programs for females in science. So I was really supported in the so-called TÜV program, supporting females on an academic track to habilitation and getting a professorship. Also with regard to funding, but also mentoring, learning all these skills and knowledge that you need to do an academic career. There, the University of Tübingen really provides the perfect environment.
We have one last question, actually, and this is, what is the key to your research?
I think the key is a really strong will to develop novel treatments for cancer patients, because I see this high need every day as a clinician in the hospital. Of course, I have this personalized experience with cancer, and I think this drives me every day to performing better and providing novel options, because it's really hard to enter a patient room and tell the patient we have nothing more to give. And I really hope that there will be a time point where I don't have to say this again to a cancer patient.
Professor Walz, thank you for being here today. This was Key to My Research, a podcast produced by Changing Time in cooperation with the University of Tübingen. You can find more information and the links to the sources in the show notes below. To get to know more fascinating scientists, please subscribe to the podcast, leave a review, and recommend it to your friends and colleagues. My name is Jennifer, I'm your host. Thank you for listening and see you next time.